My name is Artemis, and today I'm going to be presenting my biomedical engineering senior design Keystone presentation, which I completed with my partner, Maida Shaker, and we created a high-throughput in vitro model to assess paracrine factors in cardiac fibrosis. So every time somebody has a heart attack, also called a myocardial infarction, the heart undergoes a stress response when it's rebuilding tissue that's called fibrosis. And when fibrosis occurs, the tissue that's rebuilt in the heart is very stiff tissue that's rich in ECM or extracellular matrix proteins, including collagen and fibronectin. So you can actually think about that as if you get a really deep cut on your skin, and then your skin rebuilds into a really stiff scar that might stay there for the rest of your life. The same thing actually happens in the heart. So as you can see here, this is what the heart would look like after such a response. The blue is all the stiff tissue. And the stiff tissue, as you can probably imagine, actually prevents the heart from beating correctly. So that can cause issues like arrhythmias, or it's also the primary cause of all end-stage heart failure. And these diseases contribute to around 800,000 deaths per year. So this is why we wanted to look at this project, because there's no current therapy to reverse this. The only option to completely solve this problem is a heart transplant, which is very difficult to come by and very invasive. So we wanted to understand exactly how this process happens in order to, in the future, be able to build a treatment base for it. So this process, fibrosis, is mediated by cells called fibroblasts, and after an injury or a disease, such as a heart attack, they become activated, and they're activated by several paracrine signaling factors, which are just signaling pathways in the body. So this would be a normal fibroblast that's not activated in a situation where there's no injury or disease, and after injury or disease, they become activated, and these lines show the contribution that they make to the stiff tissue, so that's how they generate it. And there's a protein called transforming growth factor beta, or TGF-beta, as I'll refer to it for the rest of the presentation, which is the paracrine factor that is mostly known in literature to directly contribute to fibrosis. And there's two other factors called fibroblast growth factor, EGF, and epidermal growth factor, FGF, that also contribute to fibrosis. But it's not well known or well described at all in literature how TGF-beta works with these other factors to contribute to the heart in the fibrotic response. So that's what we wanted to study specifically in this process. And we wanted to look at the two main aspects of fibrosis, which I mentioned, which are the increased fibroblast proliferation. So these are the cells that actually contribute to fibrosis, and also the ECM deposition, which is what makes the tissue stiff. So we created a 2D cell environment to study when the fibroblast cells are activated, and we created a 3D microtissue model, which I'll explain in a minute, to study the extracellular matrix deposition. So we had two main aims in our project. The first one included looking at all of the paracrine factors I mentioned, both alone and in combination, and how they affect fibrosis in both our 2D model and our 3D model. And then we wanted to actually incorporate the cells that make the heart contract, called cardiomyocytes, and look at the effects of the contractility of the heart, which is the main concern, because fibrosis makes the heart not be as contractile. And this is our experiments that we conducted. The first is EDU staining. It shows us which cells are alive, so we use it to see, after the addition of our paracrine factors, which cells actually remain alive. And then we wanted to look further into this by actually tracking how long the cells spend in each phase of the cell cycle. So we put a virus into the cells, which acts as a sensor, and it tells us the exact amount of hours that each cell spends in the cycle of the cell cycle, so that we can see how the paracrine factors affect the actual cell cycle kinetics and how they affect how the cells operate in a fibrotic response. And then we built our 3D tissue on these devices that we have that are made up of two pillars. So we build the tissue at the top. So this is a fibrotic tissue. We added the paracrine factors, and we can actually see how much force the stiff fibrotic tissue exerts on the pillars. And that's actually going to cause the pillars to curve inward, and we can measure that force. Finally, as I mentioned, we added the cardiomyocytes to look at the same thing, but with the contractility of the heart. So here are some selected results. The first is from our EDU assay. So here we see, in the presence of paracrine factors, how are these cells activated and how do they proliferate? And we found some really interesting results, that TGF-beta alone, as you can see here and here, does not activate a significant amount of fibroblasts, so there's not a significant amount of fibroblasts proliferating. However, when we combine it with FGF or EGF, the other paracrine factors, it causes a very significant increase in fibroblasts proliferating, so it causes a much more significant fibrotic response. And the same results we found in our Fuji data, which is the sensor that tracks the cell cycle phases. So this was in the presence of TGF and EGF, we're able to see distinct phases in the cell cycle by these peaks here. And actually, if you look past the dotted line, you can see that the cells take two different paths. So this means that the cell is actually dividing. It's undergoing mitosis, which is a healthy cell function. However, in our serum-starved control, which doesn't include any growth factors to contribute to fibrosis, we're not seeing any specific changes. In fact, these peaks are most likely just noise because they only last about an hour. And we also don't see any mitosis. So these cells are not healthy. Without the growth factors, the fibroblasts are not healthy, so they're not able to contribute to any fibrotic response. And then we looked at our tissues. I included some pictures here so you can better understand this. So on day 0, we have our serum-starved cells, which again have no growth factor, and the EGF and TGF-beta. And these are the two pillars, so the distance between the pillars is the same. But then on day 6, we see that our serum-starved control, there's a tissue formed right here. This is the tissue, but it's not actually pulling on the pillars at all. However, on day 6 of our EGF and TGF-beta, if you look really closely here, you can see that this pillar is moved inward, and the distance between this and this is smaller. So this line that is at the center of this pillar isn't at the center of this one. It would be here. Sorry, the laser point is kind of shaky, but so that means that the stiff fibrotic tissue is exerting force on the pillars and pulling them inwards. And if we quantify this data, we see very similar results with our 2D assays. So EGF alone, or the growth factors alone, don't really have a significant effect on the tension, so how much tissue is pulling on the pillars. But when we have this TGF-beta and EGF condition with the growth factors working in synergy, they're able to produce a significant fibrotic response. And so we created this model. It can be used to study these growth factors in both 2D and 3D, but it can also be used to study all different kinds of fibrosis. There's liver fibrosis. It can be applied to many different growth factors as well. And we hope in the future that a model like this can be used to better understand specific fibrotic processes and then be used to inhibit these pathways from occurring. So we can inhibit these growth factors because we know their source and we know how they contribute to fibrosis, and then hopefully contribute to a therapy so that organ transplantation is not the only solution. And I would also like to thank everyone in the Chen Lab at BU, especially our advisors, Chris Chen, Jeroen Aikmans, Jordan Ewald, and Anish Vasan, and my partner for helping us with this project. And I'm happy to take any questions.
Thank you so much for your presentation. I found it so interesting. I'm curious to learn a little bit more about those pillars. Is it what is it made out of? And is it trying to imitate something that's inside of the body, or what's the story?
Yeah, that's a great question. So the pillars we make to seed these tissues are not supposed to imitate what's inside of the body, because a tissue in itself is a group of cells that are working to contribute to some sort of function. So that actually imitates enough of what's going on in the body because the cells are communicating with each other. The pillars you can think of as kind of the material resembles kind of like a hard putty. So in the beginning, it's a liquid that we put in the oven and then it hardens, but it's hard enough that the tissue can form on it without the pillars breaking, but it's soft enough that when the tissue gets really stiff, it can pull on the pillars and they can move inward because of that tension force that I was talking about. So yeah, kind of like a hard putty, and it's not supposed to recapitulate anything. It's just made from a polymer called PDMS.
Thank you.
Sure.
Thank you again for your presentation. I had a quick question if you were able to look at any genetic diseases that would cause an overproliferation of some of these types of cells, and if those would also be used in experiments like these for treating a disease that's not necessarily just a scar tissue.
Yeah, that's a really, really great question, and I think that's a huge part of the problem. We're not able to look at that in this specific project, but I think that's why we wanted to focus so much on the fact that this is a model that you can use in both 2D and 3D environments. So and all of the cells that we use are primary cells, so we get our cardiac fibroblasts from real patients. So if you could, obtained cells to either make the 2D assay that is just cells or the tissue from a patient that has some sort of genetic predisposition, you could very easily use this model, test the same things, and see how there's a difference when somebody has a genetic disease.
Great job. So I was just curious, like, in general, things in the body happen for a reason. So are there any upsides to fibrosis and therefore any risks to disabling fibrosis in the future with any treatments for this type of thing?
Yeah, that's a really great question. So fibrosis occurs all the time in your body. As I mentioned, it can be good. Like if you have a cut, the process in the cut healing is fibrosis. This is specifically for this fibrosis that occurs in people that have had a very traumatic injury or a disease, such as a heart attack, that the body tends to overcompensate and overestimate, and that's what causes these. Also, it can happen a lot in people who have these things happen several times. So if somebody has like a second heart attack, then they're more likely to experience a fibrotic response of this extent. And so all of the therapies that we're looking at are for a fibrotic response that is more than the average and would cause you health complications. So more likely these therapies would be given to someone who either has a predisposition, like Emma was saying, or has had something very traumatic happen to them and is likely, based on previous data, to experience this. Okay, thank you so much, everyone, for listening.